share my screen. Can everyone see the screen?
Yep.
Yeah. Okay, so I'm going to talk about changing blood types in donor kidneys as a solution to try and expand the transplant access. So one of the big problems in transplantation is the shortage of organs. So this graph is taken from NHSBT, and it's showing us the deceased donor kidney programme over the last 10 years. So the boxes in blue at the bottom are the number of donors each year, and in grey is the number of transplants that we perform. And the line represents the transplant waiting list. And you can see since really 2020, there's been an increase in the number of people waiting for a transplant, and currently there's just over 6,000 people waiting for a kidney transplant. So that's really the highest level ever in the last 10 years. Now, in order for a kidney transplant to be successful, it's really important to match the blood groups to have a compatible transplant. So there's 4 different types of blood groups: type O, type A, B, and type AB. Now you can see that type A and B have these little flags on the surface of a cell, or these are called antigens, and they depict what type of blood group we are. So type A has type A, type B, B, and type B has both a combination of A and B. And you see that if you're a blood group O, that you don't have any of these on the cell surface. Now, each of us have antibodies to the opposing blood group, and these are called antibodies. So if you're a blood group O person, you'll have anti-A and anti-B. If you're blood group A, you'll have an anti-B. If you're blood group type B, you'll have anti-A. And if you have blood type AB, you have no antibodies. So what this means in terms of transplantation, and if you just focus on the right-hand column, the recipient blood group. So if you're a patient needing a kidney transplant, you can, and your blood group type O, you can receive a blood group O kidney from a blood group O donor only. But if you're type A, you can receive from a type O and a type A donor. And if you're type B, you can receive from O and B. And if you're type AB, you can receive a kidney transplant from any blood group donor. So what this means in terms of transplantation is that there's a huge disparity in access to kidney transplantation. And what this graph shows us is that if you're a blood group O or B, you'll wait almost twice as long to get a kidney transplant compared to if you're A or AB. And this is a particular problem for our Black and Asian population, where 30% of them are blood group B, and they represent about 35% of the kidney transplant waiting list. So really, we've been trying to solve this problem, and first do that, let's just go into a little bit of background about the biochemical structure of these blood group antigens. So if you have blood group O antigen, or you have an H antigen on that cell surface, that's represented at the top. If you're blood group A, you have an A antigen, and a blood group B, you have this B antigen. You can see here by the depicted on the blue square on the A antigen and the circle on the B. So in recent times, enzymes have been discovered that are derived from bacteria that actually can remove these blood group antigens. So there's 2 that can remove blood group A and 2 that can remove blood group B antigens. So this would be a very good way in converting a blood group A or AB or an AB kidney into a blood group O kidney. So this would mean it would be a universal donor blood group, so it could be transplanted to any of recipients. So these blood group antigens are expressed on, as I said, on the cell surface of red blood cells, but also on the cell surface of cells that are lining our blood vessels, which are called endothelial cells. And these also line vessels within the kidney themselves. So this is a representative really of where these blood group antigens are. So we have the B antigen. So you can see the nice pink circles. So this is the endothelial cells with the expressing the B antigen. And that's just here represented here. So in order to really try and think about converting a blood group A or B kidney into a blood group O kidney, we have to think about how we're going to give these enzymes. Are we going to treat the patient? Well, this would be quite difficult. If you give the enzymes to the patient, we wouldn't guarantee that they would directly target the kidney, and they may have unwanted side effects. So it would be much better really to treat the kidney directly, so administering it to the kidney while it's on a machine. And there's different ways we can do this. So these machines are designed to circulate preservation solution through the kidney. So the kidney, meaning the kidney could be treated from the time that it's donated after and then until the time it's transplanted. And we can use machines that do this by circulating cold preservation through the kidney. So we call this cold machine perfusion, and this means that a kidney can be put on a machine, then transported from the donor to the recipient centre and be treated with an enzyme circulating in the solution that's going through the kidney. These machines are used throughout Europe. There's a number of commercially available machines, so it will be very easy to do. But we can also treat the kidney using a different type of machine perfusion called normothermic machine perfusion, or warm machine perfusion, and this involves warming the kidney up to a normal body temperature and circulating again a solution which is oxygenated. And in this way the kidney maintains its function and we can introduce an enzyme. So what we've done in the lab really is looked at trying to deliver these enzymes with both warm perfusion and during cold machine perfusion as well. So the first set of experiments we did was really to take blood group A donor kidneys and try and convert them to blood group O. So we're very fortunate in the lab. So sometimes kidneys that are donated for transplantation are not able to be used for many different reasons, but they're deemed to be unsuitable. And we have approval to really use these kidneys for research. So what we did in this set of experiments was to take a pair of kidneys from a blood group A donor. One served as a control, and then the other one was treated. So in the treated kidney, we treated it with the enzymes that can remove these A antigens. So in the first panel here, if you look on the left-hand side, you can see the control kidney, and this is a time course if you work your way down. So you can see the nice pink ring, which is our A antigen on the surface of these endothelial cells, and it's there in the treated one as well. And this is before we've done anything. And you can see after 1 hour of treatment, in the control kidney, the A antigen is still there, so we still have the nice pink colour. But in the treated kidney, you can see that there's the greens appeared. So this is the A antigen has gone and the H antigen is expressed, meaning that we've converted this into an O type kidney. And again, it's there at 6 hours. We did a similar set of experiments in introducing the enzymes during cold machine perfusion. Again, it works in the same way, but maybe just a little bit slower. So after 1 hour, we're still seeing some of the A antigen, but after 2 hours, that A antigen is gone. So we've shown in the lab that you can successfully convert an A kidney into an O kidney, but what would that mean if you transplanted it? Would it be successful? Could we do an incompatible transplant? So what we've done is model this in the lab by again taking a pair of A kidneys and then treating one to convert them to O and having one as control. After the treatment, we've then added in these antibodies into the circulation. So if you have an incompatible transplant, these antibodies will stick onto the cell surface of these endothelial cells and they would cause an immediate rejection. And what we see, so if you look at the first panel on your left-hand side, you can see that these antibodies have actually stuck to the endothelium, so we get this nice bright yellow staining, whereas in the treated kidney, there's no expression, meaning the antibodies haven't bound to the endothelium. And we can look at these antibodies in the circulation. So in blue, we have the control group, and we see a gradual decrease of the levels of antibodies, meaning they have actually stuck to the cell surface on these endothelial cells, whereas in the treated, the antibodies are still circulating. We've extended this work to look at a really rare blood type called the Bombay phenotype. So this is a population of people that have no H antigen, so they can't receive a transplant from any blood group, including blood group O. So again, this is quite rare in this country, but the prevalence of it in India is about 1 in 10,000. Again, there's been an enzyme formulated from a bacteria called Fuco-B that can actually remove that H antigen. And we've done a very similar set of experiments in that we've taken human kidneys of blood group O, and then we've treated them with the enzymes during normothermic machine perfusion or warm perfusion. And what we find, so you see on the left-hand side, this is before treatment. So you see the nice green bright staining, which is the H antigen. And then after 2 hours of treatment, it's completely gone. So it works very, very quickly. So we've shown that you can remove these blood group antigens, and that if you then were to transplant it, that it would be in the immediate phase, it would be a successful transplant. But we know that what would happen is that these antigens will actually re-emerge within the kidney, and this may cause problems. But we don't know when these antigens will actually re-emerge. So the next set of experiments was to try and determine that in the lab. So we took some of these endothelial cells from an O-type donor, and we treated them with the fuco-B enzyme. We then cultured them for 7 days. And what we found is that if you look at the line on the top, that just basically tells us that these cells are viable out to 7 days in a culture. And then if you look at the line on the bottom, you'll see this is the H antigen expression. So we start off at time 0, so this is when the cells have been treated, so we have no H antigen. But within 2 days, this H antigen is starting to be re-expressed to about 40% of what we would see normal. So not fully re-emerged, and that really has remained stable for 7 days post-transplant. So we know that this gives us a really good window for a successful transplant, and that maybe the patient might need some further therapies in between days 1 and 2 to mean that the transplant would then not reject after this time. So really, we've shown that this blood group conversion is a very simple and feasible strategy to try and increase the availability of donor organs. The action time of the enzyme is very short, between, well, actually 1 to 6 hours. We've got the machines that we can use to deliver these enzymes, and we know that we can really avoid this hyperacute mediated rejection response in the models we have in the lab. So we think that, and this strategy is very simple to modify to access for rarer blood groups like the Bombay type. And I think we've got a lot more work to do to really show that this can transform patients, really. But this is a very exciting opportunity for the future. Thank you.
Thank you, Sarah. Really interesting work going on at Cambridge. So yeah, it was good to see, and lots of lovely staining images in there.
Thank you.
If I could just encourage anyone that's got any questions to pop them into the Q&A function, and I will now hand over to George.
Good evening, everyone. I'm really sorry. I'm struggling a bit with these slides, and let me just try and attach them again. There you go.
There we go.
Perfect. I do apologize. So I'm George. I'm a surgical registrar in the Northwest in Liverpool, and a PhD candidate in bioinformatics at the University of Liverpool. These are my disclosures. I don't have any conflicts of interest related to this presentation. I don't want to be preaching to the choir, but this is probably one of my favorite sets of slides, because this is a report that's very dear to me, as it kind of forms the basis of what I'm doing. So Kidney Research UK have published this in June 2023, and this report is called a UK Public Health Emergency. It kind of shows us how big and how important chronic kidney disease is. We know it's projected to become the 5th leading cause of death by 2040. And apart from the impact that has on our patients and their families, it also carries a big total UK economic burden that exceeds £7 billion per patient per year. We've got around 30,000 patients on dialysis at present, and as a UK consortium, we perform around 3,000 kidney transplants per year. And this is the big problem, is that this number is projected to quadruple in the next decade. Our colleague Hannah Burson from Guy's and St. Thomas did a large registry analysis on over 20,000 kidney transplant recipients from the UK. in 2019. And we know that rejection remains an important problem, with about 1 in 5 of our patients losing their grafts prematurely within the first 5 years post-transplantation, one of the leading causes being rejection. What we do at the moment, in terms of diagnosing rejection, relies on pretty much a biomarker that's been described a century ago, and it culminates with an invasive test, which is a biopsy. And I'll get to that in a second. But the bottom line is that it's a very passive and reactive approach, and it's invasive, and it lacks standardization. What happens at present in all the transplant units is we see patients like yourselves coming through the clinic, and we look at your creatinine. And whenever we see an increase in the creatinine, that kind of raises some red flags. And then we start throwing a battery of tests on you, some of them being non-invasive, or minimally invasive, because it's still a blood test. So we look at your renal profile, we look at your tacrolimus levels, we test the urine for infection, we test your urine for protein leak. We do an ultrasound to make sure that the kidney is perfusing okay, and then kind of culminates with looking for antibodies. And when we don't have the answer, then we move on to doing a biopsy. Now, biopsies, some of you would have been through this procedure, and apart from the fact that it's a horrible experience to the patient, it's very operator dependent, it's very costly. And there's been a study published by Colin Geddes from Glasgow. This procedure that carries an overall major complication rate of about 10%. Some of these complications are really, really bad, meaning that you may end up hospitalized for more than 1 day. You may end up needing a blood transfusion, which again will add insult to injury in terms of getting a second transplant, probably in the future. You may need further interventions. You may need even to be taken to theaters to control the bleeding. And apart from this, as I said earlier, it's almost like going to a murder scene and looking at the blood, the knife, and the victim, and wondering how did we get here. We rely on this biomarker that was first introduced in 1914. So perhaps there is a time to kind of move on and do better, because what we know is that the treatment efficacy for rejection decreases with time. And what we do at the moment is look at injury after it happens. So the question is, can we do a blood test that tells us whether you're rejecting your kidney earlier, to provide us a window of opportunity to treat you. So that's where we've come into play. So we've done a patient public involvement survey that was in August 23 when I started my research, and about 91% of those of you who responded said, George, if you're going to do something, look at something that looks at rejection, how to make these kidneys last longer. So we've come up with the HLA-AB study, which has got 3 arms. So first, we're trying to understand whether there are any pre-transplant factors that lead into you developing antibodies against your donor. And then we want to see whether there's anything that we can do to bring equity in the older age group of patients, because we know that the allocation scheme has changed in 2019, meaning that if you're older, you're more likely to be matched with a kidney that's is of inferior quality. And then we had this proof of concept study looking at a new blood test called donor-derived cell-free DNA, which first tested in the subgroup of patients that are very high risk of developing rejection. And that's what Kidney Research UK has funded me for, being my 3rd year of PhD. And together with our colleagues from the University of Liverpool, from the computational biology facility, we want to try and develop a tool to re-stratify our patients in terms of their risk for rejection, and see whether we can improve the monitoring and get rid of this blanket approach that we do nowadays in most of the units. And this is where we are now. I've recruited about 84% of our patients, so about 216 patients recruited in the large cohort study, and we want to do a subgroup analysis on those who are deemed high risk. So, in other words, they've got a higher antibody burden, they've been retransplanted, or they got a high level mismatch. So what is this donor-derived cell-free DNA? Well, the theory is that whenever you get injury in the transplanted kidney, fragments of DNA that pertain to that kidney and hence to the donor will circulate through your bloodstream, and then we can detect this with a blood test. The manufacturers of this assay studied the 1000 Genome Project. I don't know if you remember, this was a big publication in 2015 in Nature. They've studied around 3.6 million insertion of deletions of DNA from around almost 3,000 individuals from 20 odd population groups. And basically what these are, you can see them marked. So these are our chromosomes. You can see them marked with this orange color. And these are more or less our genetic fingerprints, if you want. So it's how we differ between each other. And that's how we can tell which DNA belongs to you and which DNA belongs to the organ that is of interest. So just bear with me. I'll try and take you through the workflow. The theory is that you get antibody-mediated rejection, and then your kidney suffers, is injured, and then DNA is shed from that kidney into your bloodstream. And then what we do at that point in time is we've got your DNA stored in the freezer, and we've got the donor DNA stored in the freezer as well. And we've got a license from NHSBT to test this and screen it. And when you come to clinic, we take a blood test from you, which is about 10 milliliters of blood, so an extra blood bottle. And then there's a series of spinning and extraction. And then to make sure that we've got the right purity of DNA, we take the samples over to the Center for Genomic Research at the university, and they test us. They tell us what the purity and the concentration is. And we run it overnight on an NGS machine. And then the next day we can put it into a software. And there you go, we've got the results. So it takes 24 hours, as opposed to a biopsy, which will take you about 2 weeks. And that's if you've got an adequate sample. And you can see these little snowflakes. Those are points where you can freeze everything just to optimize reagent usage. And we've been the first center in the UK to pilot this, and the second in Europe after Switzerland. And I'm just getting ready to test the third batch of patients middle of January. And what we've noticed so far is pretty much in line with the literature. So our protocol, you come through the clinic regularly, and we test you at 6 months and 12 months to see whether there's any difference longitudinally between the two time points. We don't test it very early because in the first 2 weeks you've just had your operation, so your DNA, your donor-derived cell-free DNA level will be high. And also in our population group that is deemed high risk for transplant, they get a special induction immunosuppression called Campath or alemtuzumab. It takes about 6 months for your immune system to properly recover, which is why we've established the testing point at 6 months. To start with, this is what the Center for Genomic Research gives us. So this is an example of purities of DNA. So they can tell us whether there's any contamination or whether this cell-free DNA is pure enough, and we need a number of over 70% to be able to make a good quality judgment. And this is how the software looks like. And this is a real example from our cohort. We've detected 0.14% in this particular patient. And you've got these markers, these indel markers that we talked about, that some of them are informative and some are uninformative. The informative ones are the ones that differ between the donor and recipient, between yourself and your donor, and you only need two to make a decision. And if you're not happy with the number of reads or the quality of one of the markers, you can exclude them from the calculation. The software gives you an average percentage of cell-free DNA that is in your bloodstream. And what we know is that, and what we noticed so far is that Whenever you get a level of less than 0.5%, that means that your kidney is functioning fine and there's no problem and there's no signs of rejection. When you start having an increased level of 0.5 to 1%, that kind of links with emergence of antibodies. So we need to start monitoring you a bit more aggressively at that point in time. And when we get a level of over 1%, we know that that is injury, and you're probably rejecting. And that happens even before your creatinine starts to take off. So for up to 2 to 3 weeks, that provides us a good window of opportunity. What I'm hoping is collaborating with our bioinformatician colleagues is that on all these data that we get from this research study, is we can come up with a tool to re-stratify our patients, because we all fall into different categories of risks. I may, for example, have never had a blood transfusion, never had a transplant, and I get my first transplant. Therefore, I'm at a lower risk of rejecting. So perhaps doing a donor-derived cell-free DNA test once a year will suffice. But for somebody who's had numerous blood transfusion, maybe 1 or 2 or 3 transplants, they fall at a very high category of risk for rejection. So maybe monitoring donor-derived cell-free DNA in the first month would be beneficial. And if that's positive, then that would trigger a biopsy. And by the time the biopsy results come back, you can already start treating rejection. And conversely, if the donor-derived cell-free DNA is negative, then you know your patient is not rejecting, and it acts as a reassurance, and it spares them from an unnecessary test, which carries a lot of complications. And these are some of the pictures. The last 2 years and a half has been a beautiful journey, lots of training, and traveled here and there trying to accelerate the implementation of this test, and seeking collaboration with some of our colleagues from across the continents and across the units. And I just want to leave you with this quote. Dr. Joseph Murray was, of course, many of you will know, he led the first successful human kidney transplant team in 1954 in Peter Bryan Hospital in Boston, USA. And it kind of reminds me that we need to remain humble, because we do witness human nature in the raw in our profession. But also we need to stay alert, because that's the only way to find new problems to solve and new paths to investigate. And this was his speech from 1990, when he won the Nobel Prize in Physiology and Medicine for his contribution to transplant. So of course I wouldn't be able to do anything alone, and there's a lot of people I have to acknowledge, and especially Kidney Research UK and the Stony Gate Trust for funding my last year of PhD, but also all our patients and their families and their donors, because we wouldn't be able to do anything without you. Thank you.
Thank you, George, and thanks for sharing, some insights into your results with us. And it was really interesting to see the workflow, the way you presented that as well. So thank you. Just in the interest of time, I'll pass straight on to Katrina.
Cool. So hi everyone, my name's Katrina. I'm a kidney doctor in London, currently working towards my PhD, and my project is exploring whether we can prevent the development of harmful antibodies by improving blood transfusion methods. So, Can I just check you can still see, because a weird message popped up?
Yeah, we can still see.
Cool. So in the next 10 minutes, I'm going to cover some general background, a little bit about the national programme, and then some details about the ongoing project and a few early results from it. So just starting with some background to the work and why it matters. So this project all relates to the human leukocyte antigen, or HLA system, and this is a major part of the immune system and helps to protect us from infection. So a bit like Sarah was talking about earlier, almost every cell in the body has HLA proteins, and these stick out from the surface like those blood antigens. And these tell the immune system that the cell is part of the body. It's sort of like an ID card. So if there's a cell present which has different HLA proteins on its surface, the immune system will spot that they don't fit the expected pattern. And the immune system then produces antibodies against the HLA protein, and these mark this different cell to be destroyed. and these antibodies are known as HLA antibodies. So this is a really useful system to spot infections like bacteria, but the same system is also going to recognize that a transplant kidney comes from someone else, and the body then forms HLA antibodies which can damage the new kidney. So there are loads of studies which show how these HLA antibodies can affect outcomes. So some of the studies are like this one. They show that patients with HLA antibodies wait longer before they receive a transplant. So on this graph, as we move from left to right, we see that the individuals with more HLA antibodies are waiting more days to receive a transplant because that black line is getting higher. And this happens because we don't offer a kidney to a patient if they've already developed HLA antibodies, which could damage the kidney. The HLA antibodies which directly target a kidney transplant are known as donor-specific antibodies. And there are lots of studies like this one, which have shown that transplant patients with donor-specific antibodies are likely to have their transplant last less long. And in this figure, these patients with donor-specific antibodies are the green line, and it goes down faster than the black line. And this means that more patients lost their transplant. And we see similar results even when the antibodies don't directly target the transplant. So a person might develop HLA antibodies anytime they come into contact with cells which are not their own. And this happens during pregnancy, when someone receives a transplant or a transfusion, or rarely as a result of infection. And once HLA antibodies develop, it's very difficult to completely get rid of them again. So in kidney transplantation, we match the HLA proteins between the donor and the recipient to try and reduce the number of HLA antibodies which develop, as this has been shown to lower but not remove the likelihood of rejection. And as part of this project, we're just starting to do a similar sort of matching for blood transfusions for patients who need or who have a kidney transplant. So the next couple of slides are just giving an overview of the national programme. So this started as a collaboration between NHS Blood and Transplant and Imperial College Healthcare, and it's now being rolled out to different transplant centres. At the moment, it's available at 3 transplant units. The Royal Free and Newcastle were added in this year, and there's a timetable to bring all of the transplant centres into the programme over the next couple of years. HLA matched blood units are being offered to patients who are on the transplant waiting list and patients with a functioning kidney transplant. And so far, at Imperial, we've given 220 units of blood to 91 different patients. And so finally, I'm going to go through the actual research I'm doing alongside helping with this delivery of the national programme. So the national programme is the first time where HLA selection of blood has been done across a population. Because of this, we don't know what the best way to select blood will be. Currently, what we're going on is based on what we know from matching transplants for HLA, but it's likely there'll be some differences for a number of reasons. My research is trying to work out what would be the best way to select blood for transfusion to maximise transplant success. So the first part of this work has been exploring who develops HLA antibodies, because this helps us to make predictions about whether a future individual is likely to develop antibodies if they receive a blood transfusion. What we've seen so far is there are differences in the individuals who develop HLA antibodies. So women are more likely to develop HLA antibodies, as are younger patients. I've also found individuals from global majority backgrounds tend to develop HLA antibodies more as well. Finally, I've spotted that individuals who already had HLA antibodies before they received a blood transfusion are more likely to have new HLA antibodies after the transfusion, compared with people who had no antibodies before the transfusion. We'll need to do a bit more work to look at this in some more detail. As well as differences between the individuals, there are some differences in the blood units, which can change whether the recipient is likely to develop HLA antibodies. A longer time between when the blood was donated and when it is given reduces the likelihood of antibodies forming. And this is because the blood cells are a bit more fragile. But it also means that blood can sometimes be less good for treating low blood counts, so it's a bit of a balance. Previous studies have also shown that the blood donor's medical history can play a role in the development of other types of antibodies, and this is something we're in the process of exploring for HLA antibodies at the moment. The second part of this work is seeing whether the selection we are doing is reducing HLA antibody formation. As with organ transplants, for transfusion, we rely on blood donors to come forward. At the time when someone needs a blood transfusion, we have a limited supply of units from which we can select, so it's rare that we can achieve a perfect match. This chart is a comparison of how the selection is improving the matching. The bottom row shows the random units of blood that we were giving prior to the study, and the green block on the far left would be a perfect match, and the red block is the worst category of match. So with the selection in the top row, we're managing to give more patients perfect matches, so the green box is growing, and fewer patients are getting the worst matches, so the red box is shrinking. But we're still not managing to get a perfect match into everybody, which is what we would expect. But what we now need to understand is whether the improvements we are managing to achieve is actually leading to reductions in antibody formation, as we predict that it should. And this is work is ongoing at the moment and will hopefully be published early in the new year. And this last step of the project is going to be to bring everything together to design a system whereby the patient's details is put in at one end of the system, and the system will calculate from all of the blood units which are available across the country, which would be the best one to give to that particular patient. Whilst at the moment the study is only at a few centres, ultimately it's being rolled out across all the centres, and we need to make sure that everyone is benefiting equally from the programme. So the system will take into account all of the requests it receives on a single day and matches the best unit to each of the patients so that the benefit is equal across the country. And this project is obviously not something I'm doing in isolation, and I've had the support of these wonderful people from Imperial NHS Blood and Transplant. I'm also hugely grateful to all of the funders who make this research possible, and especially to KO UK. Thank you very much.
Thank you, Katrina. Yeah, really lovely slides and a great explanation. I can see that there's a question in the chat, but we'll address that at the end, and we'll pass over to Sam for now.
Great, thank you so much. Perfect. So hi, my name's Sam. I'm a surgeon and a PhD student in Newcastle University, and thank you very much to Kidney Research UK for the invitation to talk and for funding this research. So I'm talking today about using a special type of ultrasound scan to try and check the health or quality of kidneys that have been donated for transplantation whilst they're still outside of the body. I'll first just quickly say thank you very much to the patient and public research panel in the NIHR BTIU, who've had a real role from the beginning of this project in terms of the design of the study, planning the study, and helped with grant applications, especially Hannah Murray and Sian O'Day, who is the public co-applicant of the NIHR BTIU. So as I think everyone here knows, kidney transplantation for most people with kidney failure is the best treatment, and it offers several different advantages over and above dialysis. We heard from Sarah's talk about the use of machine perfusion, and specifically warm machine perfusion that allows us to keep kidneys alive outside of the body. And this allows us to deliver therapies, which is what Sarah was mostly talking about, but it also offers a potential with an active and alive organ to assess that organ's health and how it's going to behave once it's transplanted. And there are some kidneys where it's really obvious that they're going to work really well after transplant. Some kidneys where it's obvious that they're not going to work well and therefore we don't transplant them, but there's this big grey area in the middle of kidneys where it's not clear whether they'd work well. And by having new tools that can let us assess the health of the kidneys, it will allow us to safely transplant some kidneys that otherwise might be deemed too high risk. The way that we decided to look at the kidneys in this study was to use a special type of ultrasound. And I think we're all at least a little bit familiar with ultrasound as the scan that pregnant women get to have a check on the baby. And what we know from that is that it's a really safe type of scan that can give us an immediate result. So we put the scan probe on someone, we can immediately see what's going on. As well as being used in pregnancy, it's used in a wide range of different diseases. And we thought that it would be useful to look at kidneys outside of the body. The thing specifically that we wanted to look at was small vessel blood flow. So blood flow in these sort of really important key functional areas of the kidney. And to do this, we used a type of essentially dye, which is made up of small bubbles. And this is something which is used clinically to look at people's kidneys that are alive. And essentially, when we inject these small non-toxic bubbles, they're able to travel in the small blood vessels and they show up very brightly on the scan. And the key research question was whether this could be used as a measure of kidney quality. So I'm going to show you a quick video of what these scans look like. The panel in the middle is just how we get a good view of the kidney. The panel on the left is where all the action is. And the nice thing is that because we're outside of the body, we can place an ultrasound probe directly on the surface of the kidney. So if I press play here, you see blood flow shoot up through the kidney into this area, which is the cortex, and then down into this area, which is the medulla. And this is an example of a very healthy looking scan. What we can then do is look at each of the individual sort of frames of that video as a separate picture. We can draw around regions of interest, the areas that we're interested in, such as the cortex and medulla, and then we can extract information from that video. And here, each black dot on these plots is the information from one single image within that video. And then we can analyze all of this data together, and we can come up with metrics which tell us about how well perfused, how good the blood flow is to that area of kidney. And here's an example with 5 human kidneys, which, similar to Sarah's work, these are kidneys that had been retrieved with the intention of them being transplanted and then were deemed too unhealthy for transplant. And each of these dots represents an individual kidney. And moving from left to right on this bottom axis means that the ultrasound scan is better. It's showing better blood flow to those regions. And then moving down this axis, this is where we've looked at the kidney under the microscope. And moving down this axis means there's more dead cells. And what you can see is that the better the ultrasound is, it tells us that there are more living, happy, healthy cells in the kidney. We also looked at the ultrasound with a marker of injury, which is a marker that can be used clinically. And then we found that doing a scan at just 2 hours, if we then continued that kidney alive outside the body for a further day, it predicted how much injury there was a day after the scan was performed, which gave us some confidence that this might be able to predict what happens to a kidney after transplant. Another thing, again, that was highlighted by Sarah was there's lots of exciting new therapies that we're able to deliver during machine perfusion, such as stem cell-based therapies and other advanced kind of gene editing therapies. And one thing that we need is a way that we can monitor response to these therapies. And we've shown here that contrast-enhanced ultrasound is able to do that. So this is data from a pair of kidneys. The kidney that I'm going to show on the left had a cell therapy treatment to try and improve its quality, and the kidney on the right was a control kidney. And if I play these both at the same time, you'll see that the blood flow to these important areas of the kidney when the cell therapy is given is really, really good, and this sort of mirrors what we saw on the healthy scan previously, whereas the control kidney has very, very little blood flow to these main areas. So it's not just able to take a kidney and tell us what the quality of that kidney is, it's also able to be used in real time and tell us how a kidney is responding to a specific treatment that we're delivering to it. We really wanted this to be available to other people for use. So all of the methods that we developed around how to analyze these images and how to go from a scan that someone's taken to a numerical result of this is how good the quality of blood flow in this region is, we've made all of those tools available. So there's a website that other researchers can log into and use, and there are also software packages that other researchers can use on their data. So in conclusion, we've shown that this type of ultrasound scan is a safe and non-toxic way that we can get an immediate assessment of kidney health. We don't have to take blood or biopsy and process it. We just put a probe on the surface of the kidney and get an answer straight away. It can also assess the response to new treatments. We've analysed and developed different techniques for analysing these scans, and we've created free-to-use software and web applications that other people can use. And the key next step now is to perform these scans on kidneys that have actually go on to be transplanted, so that we can see how these scans correlate with how the kidney does after transplant. And we've now been awarded a clinical grant to do that work. And the overall aim of this is that we can enable the safe transplant of kidneys that otherwise would be deemed too high risk, and this would enable us to perform more life-saving transplants for people on the kidney waiting list. I'd just like to highlight again, this is a massive project where a large number of people have contributed, including the patient and public research panel from the NIHR and Kidney Research UK as well. And thank you very much for listening.
Thank you, Sam. There were really good moving images in there. Interesting to see the examples of the ultrasound. So thank you. We do have a question in the chat, so yeah, encourage everyone. I guess the Q&A function is not working, apologies. So yeah, if you do have any questions, do pop them in the chat because we can see them. So we do have a question in from Joe, I believe it's for Katrina. And the question is, what is global majority background?
So global majority background is just a term that's replaced either minority ethnic or Black and Asian minority ethnic. It typically refers to people with non-Caucasian background who represent the global majority in race.
Thank you. Yeah, it doesn't look like we've got any questions at the minute. So in the meantime, I'll just sort of ask maybe one myself, maybe to all of you. Just looking ahead maybe to the next 5-10 years, what do you think would be the biggest changes or breakthroughs in kidney transplant research?
Shall I start? I'll start. So I think, I mean, where we're heading at the minute is certainly using these machine perfusion technologies that are not currently used as standard practice in the UK. But I think the introduction of those is certainly here at the minute. So we want to see widespread use of those and then to use them as a way of delivering different therapies in to the kidneys. So one of the big problems is a lot of the kidneys that are donated now are from older donors or donors with past medical history that make them a little bit suboptimal. So we would want to try and improve the quality of these kidneys, as Sam said, you know, and use techniques like the ultrasound to make a better assessment of their quality to try and expand, you know, the organ, you know, the number of organs for transplant that we can do really. So we see that as I think definitely in 5 years we will be there. We will be putting treatments in kidneys and transplanting them successfully.
I think as well, a lot of these kidneys are kidneys that are working very, very well in the donor, but because, like Sarah says, it's an older donor or a donor with some comorbidities, we know that those kidneys, even though they're working well in the donor, will fail to tolerate the sort of injury that's inherent with transplant. So, you know, we've got to get that kidney out of the donor, the kidney's then got to be transported, and then there's the implantation, which that phase of things causes further injury to the kidney. So if we can have new therapies, like Sarah says, that helps prevent some of that injury and helps the kidney to recover quickly from that injury, which is inevitable, and get it back to a working organ like it was in the donor, that opens up the door to lots more organs that currently just aren't of a high enough quality to be used.
Thank you.
I think in a way I'm echoing, I think a lot of the work's going to come in improving the organs which are already donated to reduce the number that end up not being used, whether that's through treatments, through understanding actually these organs are of better quality than we think they are, all of which is just going to increase the number of donor organs available, and so the number of people transplanted.
Yeah, I mean, there's definitely a push to increase organ utilization and decrease the discard rate. And there's a lot of work, as you know, Sarah and Sam and Katrina were saying, in the pre-transplant period. But also from my perspective, my research interest is mostly what happens in post-transplant immunosuppression. I think we will see an implementation of this blood test that we're working on in the next 5 years, and that will change the way we select patients for biopsies, and maybe we will start seeing more personalized medicine and tailored immunosuppression to each individual case. So there's a lot of work happening in the post-transplant phase as well.
Thank you. Thank you all. Another question has popped in the chat, and it's about the blood transfusion work. So Katrina, is this currently done in Oxford, and how long until this might be a standard process?
So on the first bit, not yet. At the moment, it's just Imperial Royal Free in Newcastle. There is already a schedule for when each of the transplant units is brought online. I was just trying to look it up so I could see where Oxford was on that. And it's all over in the next 2 years. It's bringing all the centres online. And So hopefully by the end of that, it'll then be standard. And then from the transplant centres, it will go out to the satellite units, eventually all the units.
Thank you. And then one more question, I guess, can be directed to who would like to pick it up. But the impact of this work limited significantly by the ability of units to work with offered organs due to system staffing, theatre availability pressures.
Yeah.
I think all of those questions are massive problems that we could speak, take an hour speaking about. There is now a pledge to funding assessment and reconditioning centre pilots. I know Sarah's really involved with it. I think George is involved with it also. And essentially, these are hubs where organs will be sent to, and it'll allow us to centralise some expertise and relieve some of those staffing pressures. And the aim of those hubs is to have the type of organs that we're speaking about go to a centralised hub so that we have everything there to deliver these kind of therapies and deliver these viability assessments, and then the organs would go out. So that is an area that is sort of concerning everyone, and there are things being put in place to try and overcome those hurdles. But they're not easy hurdles to overcome.
For me, the other thing to mention is I get exactly when I think it's in March coming next year, they're changing how the offering is being done to try and shift more of the operations into the daytime and more of the retrievals into the nighttime to actually maximize the utilization, so that in order to try and increase capacity across all the transplant units.
Thank you. Thanks for answering that. Yeah, if there's no other questions, we have reached the end of the webinar. Thank you everyone for joining us. Thank you for the researchers for joining us and sharing your work with us and for answering the questions. For everyone to know, we will have another webinar in the new year. We're just in the middle of finalising a date, but we will be sharing this on socials and in other channels. So do keep an eye out, and yeah, thank you all for joining us.
Thank you very
Thank you so
much.
much.
Thank you. Thank you.